This method permits the transcriptomic analysis of a rare cell population from Drosophila male accessory gland called the secondary cells, and it uncovers the genes that make them critical for reproductive success. In the course of one day, this technique allows the sorting of secondary cells from different genotypes and the extraction of their total RNA for subsequent qPCR or RNA sequencing.
Sometimes projects are limited by the rarity of starting material. This method can provide the basis for other groups facing the challenge of working with a rare cell population.
Because the procedure needs to cumulate 40 glands, it's important to practice the dissections. Preparing the homemade pipette tips is also important, and so you should visualize it on the video how to make it and prepare in advance.
Before beginning the procedure, cut and flame round, wide 200 microliter tips for handling the glands, and pass the tip opening of multiple 1000 microliter tips near a flame for less than 1 second to narrow the openings. Then sort the modified tips from narrower to wider based on their speed of aspiration. For accessory gland dissection, place 20 to 25 male Drosophila in a glass dish on ice, and use forceps and a dissection microscope to remove the reproductive tract of one male fly. Clear the accessory gland pair from all of the other tissues, including the testes and ejaculatory bulb, and transfer the accessory glands to a glass plate containing serum-supplemented medium at room temperature. When 20 pairs of accessory glands have been collected, wash the